Okay, our next speaker is Bailey Howell.
Okay, no. Yeah.
Okay, so my name's Bailey Howell, and my project is on the effects of urbanization on topaz shape and lamellae size in Anole chrysatilis. So this is Anole chrysatilis over here. There we go. So Greater Antillean anoles are evolutionary model organisms. And on these islands in the Greater Antilles, you see repeated examples of convergent evolution for these ecomorphs. So basically, on different islands, you have distantly related species that fall into these same categories based on how they partition their habitat, so where they live in the tree. And another interesting thing is greater perch heights have been correlated with greater topad area and greater lamellae number. Lamellae are specialized adhesive scales. So these pictures here kind of demonstrate that. The picture at the top shows a greater topad area, and then this picture down here demonstrates a smaller topad area. So Anole chrysatilis in Puerto Rico show changes in multiple traits between urban and natural populations. Here you can see greater topad area in the urban population, smaller topad area in the natural population. Other things were lamellae number was increased in urban populations, and there were greater relative limb lengths in urban populations as well. And this is presumably an adaptation to using smoother substrates. So they need to cling, the claw is less effective, so they have more of those specialized adhesive scales, and they have greater topad area. So we know that the shape or the area of these topads is changing, but what I wanted to investigate was how the shape was changing to accommodate that change in area. So this was in Puerto Rican crested anoles and Anole chrysatilis, and there were 3 hypotheses about how this might be happening. So the very top one is an isometric change between urban and natural, and then the urban are increasing in width or the urban are increasing in length. And to determine this, I use geometric morphometrics, which is a method that uses landmark data to quantify shape, and I use scans of individual topaz. And I did statistical analyses in R using the geomorph and morpho packages, and those are basically just packages that let me work with highly dimensional shape data. So to collect the data, my collaborator, Dr. Kristen Winchell, she captured and imaged the topads of over 1,000 adult male Anole chrysatilis from 5 municipalities. I'm sorry if I butcher these, but Ponce, Aguadilla, Arecibo, Mayagüez, and San Juan, with 13 sites, 7 urban, and 6 forest. And I used around 20 individuals from each site for a total data set of 246 images. So I didn't have a total of 20 for each site, but I had as much as I could have for that site because some didn't have 20. And then for the landmarks, I added the landmarks in TPS Dig, which is software which lets you do that. This is kind of what it looks like over here when you input the landmarks. And I had landmarks that go around the shape of the entire toe, just the toe pad, and then lamellae 5 through 10. And I chose to avoid the top few lamellae, which are the purple lines, because they can be kind of small and difficult to see, and you want to be able to compare them. And so there were a total of 19 landmarks and 13 curves with 8 semi-landmarks per curve for each specimen, so all 246. So to analyze the shape data, I used the landmarks, and I performed a principal component analysis with that data. So that's a PCA, a canonical variant analysis, a CVA, and a between group principal component analysis of BGPCA. And then I used a Procrustes ANOVA, which is basically just a statistical test for highly dimensional shape data to determine if site type, urban or natural, or municipality were useful to predict shape, as well as looking for an interaction between site type and municipality. And then to take it further, I extracted linear measurements from the landmark data, and this over here is a demonstration of that. So basically, for proximal toe length, I took the average of landmarks 1 and 2 and the average of landmarks 3 and 4 and found the distance between those average points. And then same for topad length, taking the average. And then for topad width, it was landmarks 5 and 6, the distance between those. And then for lamellae size, I had to take the average of all of these points, and then that was lamellae size. So I use those linear measurements and natural log transform them to determine if there were significant differences between site type among municipalities and look for an interaction between site type and municipality there as well. So the first result we have is the PCA. So this is an individual level analysis, which is why the individuals are all color coded based on whether they're urban or natural, so that you can hopefully see differences between the groups. What the PCA does is it basically finds new axes that can explain the most variation within your data, so it doesn't necessarily have to be based on whether they're urban or natural. And these plots at the bottom demonstrate the extremes of each axis. And so for axis 1, it looks like we're looking at how bent the toe is. For axis 2, it seems like we're looking at lamella size, but that's kind of hard to see. You'll just have to take my word for it. For axis 3, though, it actually begins to look at what I was interested in, which is shape. And so basically, like, this kind of seems to be more how much relative toe pad do you have, like, for your entire toe. And it seems like there's some separation on this axis, but of course you're just eyeballing it. But it looks like the urban are shifted more towards the maximum shape there, and the natural are shifted more towards the minimum shape, minus this one funky individual up here. But we'll do more to see. So basically what I decided to do after that to actually compare groups, urban and natural, is the CVA and the BGPCA. So for the canonical variant analysis, you just give it your groups, and it is like maximize shape differences between these two groups. And so it finds the axis that does that. And so that's why we have this one axis, and they did clearly separate out based on urban in the gray and green are natural. But if you were to randomize this, you would still expect to find an axis that separates the data just because it's a two-state categorical variable. So you would see a little bit more overlap here though. But I wanted to do sort of a sanity check, like, okay, are we just picking up randomness or is this really different? So that's why I did the BGPCA. And with the BGPCA, it's just like the CVA, but it doesn't standardize by within group variation, which is where a lot of error can come in here. And so you see there is a lot more overlap here, but that's to be expected. If you were to do this with randomized data, they would be almost entirely overlapped, but actually with this analysis, you see that there is a definite shift here as well. So that was like, okay, we're picking up actual differences that are here. I'm not making things up. And so based on the Procrustes ANOVA, there was a difference between urban and natural anoles for overall shape. And this was consistent with the shape, projected shapes I got from the CVA, and then the mean shapes that I got over here for the BGPCA. And we won't spend too much time talking about this because I don't want to waste time, but for PC3, don't put too much stock in these because I basically color coded them based on the observable pattern, which was kind of difficult to see. So, you know, take that with a grain of salt. But here we go. So the interesting part, for that Procrustes ANOVA, there was also an interaction between site type and municipality for shape. And so to explore this further, I plotted the mean shapes for each municipality for urban and natural. So urban in gray and natural in green. And these were two that were really different. So Aguadilla here is like almost no change, whereas Ponce is really consistent with the overall trend and the changes that we saw. So like it seems like this is increasing more in length than in width. And that's really consistent with what I expected. And I'll find later that Aguadilla is actually just really weird. So we'll get to that with the linear measurements, which are coming up here. So I took those linear measurements, natural log transformed, the way I said earlier. And there was an interaction between site type and municipality for toe pad width, toe pad length, and proximal toe length. The only one we didn't see an interaction for is lamella size. So also site type and municipality, all of those were significant. So over here looking at the graph, we have urban on the left, natural on the right. In the black with the error bars, they're 95% confidence intervals around the mean. And then lines to connect the means for urban and natural. And then on the y-axis, you have the measurement in millimeters, and the y-axis is natural log transform. So that's why it doesn't increase in an incremented scale. But you have all the individuals displayed on these plots as well. So, but they're color-coded based on municipality. And there's lines which are also colored connecting the means from each municipality as well. And so the really interesting thing that you can see when you plot them like this is if you go over here to lamellae size or lamellae width, you see that Aguadilla is the only municipality that is going against the overall trend, which is why you don't see that interaction effect in lamellae size. And another thing you can get from these plots is that lamellae size and topad length are the most dramatic and the most consistent across municipality. Even though there are some changes in the sort of amount of decrease you see between urban and natural, it is much more variable for proximal toe length and topad width. So that's really interesting. So both the CVA and the BGPCA were consistent with one another and showed that shapes differ between urban and natural populations. And the changes were most consistent with hypothesis 3, which I demonstrated again over here. They're increasing more in length. And the linear measurement supported that as well. Topad length and lamellae size changed most dramatically and consistently across municipality between urban and natural populations. Some reasons for that, for the interaction could be there's more interaction and gene flow between populations at certain municipalities. Urban sites at some municipalities might have been established longer or just more urbanized. And then for the more dramatic increases in topad length, having longer topads might be more advantageous biomechanically. So that could be for producing cling force, or that could be for greater sprint speeds, or it just may be a holdover from increases in relative limb length that I talked about earlier. And for future studies, you could look at gene flow between populations for different municipalities, and you could look at the effects of topad shape and lamellae size on performance. So those two things I talked about earlier. These are my acknowledgments. I'd like to thank my committee, Dr. Travis Hagee, Dr. Ross Whitlam, Dr. Nicole Welch, my collaborator at Washington University St. Louis, Dr. Kristen Winchell, the honors director, Dr. Pam Whitehead, and the Ina E. Gordy Honors College. The last three are all sources of funding I had to travel to national meetings to present my work. And if you have Redbubble, my collaborator sells these stickers on Redbubble. They're super cute. You should get one. These are my references. And these are all the plots from the different municipalities if you're interested in that and you have questions about that. I know that was a lot of information, so if anybody has any questions or wants me to repeat anything, feel free to ask.
Bailey, I was just going to say, one of the things I love about your project are all the hypotheses that it launches, which is one of the hallmarks of good research. Travis, do you all have environmental data that can be correlated with the PCA scores?
So I can kind of.
For anybody, or Bailey, do you know of?
I think that Kristen has this data because she's like super, super meticulous about the way she collects data. And she has like so much information that I didn't get to use for this. But it would just be a matter of me reaching out to her and then combing through it to see if it has what I need. But I think that there is some environmental data for sure, because I know she, for one of her papers, looked at humidity and temperature in different environments and stuff like that. So I feel like she probably does have it, but Dr. Hagee, you can also answer as well.
Yeah, I'm sure she has. I know, like Bailey said, I think she has temperature stuff, but I'm sure she also has classic perch height, perch diameter, and I think she even has a measurement of texture using taking pictures and then kind of using shadows to get an estimate of texture.
Cool, because that's just begging for it.
Right.
I have a question, Julie. I'm sorry, Bailey. Bailey, how does
Hold on. Okay, I can hear you.
Is that better? Can you hear me?
Yeah.
Okay. How does this experience, where you used a lot of computational data and programs and coding probably, affect your experience as a biology major who traditionally would be out in the field working working with specimens directly. How did that affect your experience?
So I did not know whenever I changed my major to biology, because I wasn't originally a biology major, that this was something that I could do in biology. And I actually hated computers before I got into this type of thing. But I actually did a computational biology research experience outside of this. And now I've found that I really, really love computational biology in general. So it's on my trajectory for graduate school now that I'll probably be doing a lot more of this type of thing. And it's just insane to think that this is something that a biologist can do, and that I can have the best of both worlds, working in the lab on my computer 8 hours a day if I want to, or in the field for 2 full weeks or a month collecting data, and it's crazy. Yeah, so I'm really grateful.
Bailey, do you want to tell them? Everyone else has been talking about their plans after graduation. Do you want to tell them what you're doing?
So, Dr. Welch, you don't know about this, so I can tell you about this. But I got an honorable mention for the National Science Foundation Graduate Research Fellowship Program. So that was really exciting. I was sad I didn't get it, but an honorable mention is still cool. And I am going to Virginia Tech to work in a lab where I'm going to be doing a lot of computational biology, some really cool stuff in evolutionary biology still. So yeah, I'm really excited.
Congrats.
Thank you.
For graduate school, Bailey applied for 5 different labs and got 5 different offers.
Okay.
And so
You're done, Dr. Hagee. You're done.
Things we dreamed of.
That's good, Bailey. I do have a question, though.
Yeah.
So I know you're looking at different animal. Is it possible for you to imagine our toe pad? Do we actually have toe pad, human beings?
So it would be like our hands, but we don't have anything that allows us to adhere. So no is, I guess, the answer, but it's the kind of equivalent of our hands. Yeah.
Okay. Well, that kind of invalidated my question anyway. I can't go anymore.
Yeah, it
Thank
made
you.
it more complicated.
Yeah.
We have time for one last quick question if anybody has one.
Hear me? I have a question.
Yeah.
Could you explain to a non-statistician what exactly an interaction between site and municipalities, site type and municipality, that was the interaction effect?
Yes.
What the fact that you saw that effect, what exactly does that mean in real world terms?
So in real world terms, that basically means that depending on what municipality you're looking at, the change between urban and natural is different.
Oh, okay.
So you have a different, so here you can look at Arecibo, there's a huge increase in the toe pad or the proximal toe length between one municipality for urban and natural. But if you were to look at San Juan over here, there's actually the opposite effect. It decreases the proximal toe length. So that's kind of, you taught me that, but that's what the interaction means, basically.
Okay, thank you.
Mm-hmm.
Okay, well, thank you, Bailey, and thanks to for all those questions. Great presentation.